Okay, everyone, thank you very much for coming tonight. Just wanted to introduce what we do here at Science in the News a little bit. Science in the News is a graduate student organization here at Harvard University, and we're interested in communicating science to the public. So we have a spring and fall lecture series in which graduate students such as Thomas talk about their own research and about various scientific topics. We also have Science by the Pint, which is a series of events usually at the beer end, where you can sit down and have a pint and sort of mix with scientists from a specific research group to talk with them more about what they do, sort of a more informal setting. And then we also, if you look online, we have essentially a whole blog of various articles that graduate students write on different scientific topics. So please feel free to check all those out. We have a flyer outside that you're welcome to pick up at some point, either at the intermission or at the end of the talk. And it has the list of the rest of our lecture series, so you can pick that up. And if you haven't already gotten one of the handouts, we've got a bunch more, so just let us know. And then as far as specifically the lecture series, then this is our second one of the series this spring. And the format of the talk will be that it's going to be roughly a 45-minute talk broken up into sections. After the first section, we'll have a break for questions. So we ask that you please save your questions for the breaks. And then we'll have a second break for questions later. After the first break for questions, we'll also have an intermission where you can go out and mingle a bit and have some food and take a break, and then we'll come back in and finish the lecture. So with that, I will pass it off to our speaker. I hope you enjoyed the seminar.
Thank you very much, Jess. And thank you everyone for being here. My name is Thomas Graham. I'm a fifth-year graduate student in the Harvard Systems Biology program, and I work with Joe Laparo and Johannes Walter over at Harvard Medical School. We study DNA repair, and that's going to be the topic of my talk today, DNA damage and DNA repair. Here's the short one-slide synopsis of what I'm going to talk about. Your DNA is constantly being damaged in all sorts of ways, and fortunately for you, inside your cells, there are DNA repair machines that are constantly surveilling your DNA and repairing this damage. If they repair the DNA correctly, then the sequence is maintained the same as it was before, and it's good as new. But if they repair DNA incorrectly, they can induce permanent changes in the DNA, which are known as mutations. And mutations are the root cause of genetic diseases, including cancer, although mutations can also be useful in certain contexts, and I'll talk about that at the end of the talk. So should use this. So my talk is split into three parts. The first part, I'm going to give a general overview of DNA, how it's damaged, and how it's repaired. The second part, I'm going to talk about my own research. I study a type of DNA repair called end joining that repairs DNA double-strand breaks, and I've developed a way of looking at it occurring on single DNA molecules. So I'll describe how I do that. And then third, I'll describe how both your body as well as molecular biologists are sometimes able to cooperate with DNA repair machines to make targeted changes in DNA. And then as just mentioned, there's going to be a question and answer period after every part of the talk. And we're going to have a brief intermission after the first part. So if you could hold your questions until then, that would be great. One of the things that first got me interested in biology, made me first want to be a biologist when I was in high school, was thinking about this, that all of the living organisms that we see around us started their lives as a single cell, a single fertilized egg cell. So another way of thinking about this is that the information that's needed to make this long neck or these spines or this lovable face is all encoded in a single cell. There's a lot of information that's packed inside a single cell. And as we know now, the bulk of that information is stored in a molecule. That molecule, of course, is the famous DNA double helix. So first, a little bit about the structure of DNA. DNA is made of two strands that are made of units called bases There are 4 different bases. They're abbreviated as A, C, T, and G. And then there are 2 different strands of bases, and they pair together. You can think of DNA as like a digital information storage device. So just like a computer stores information as zeros and ones, DNA stores information in 4 different letters, A, T, C, and G. I'm going to draw 2 different cartoons of DNA throughout this talk. The first looks kind of like a ladder, and the second is just parallel lines. So if you see one of these, that indicates DNA. DNA has a useful property called base pairing, where whenever there's a letter G on one strand, there's always a letter C on the other strand. Wherever there's a letter A on one strand, there's always a letter T on the other strand. And this fact arises from the chemical properties of the individual bases. G interacts preferentially with C, and A interacts preferentially with T. This is useful because you have a built-in backup copy of the information in the DNA. So you have the same information in duplicate, although here there's a T and here there's an A. And a little piece of terminology, one of these pairs is referred to as a base pair. This is also useful because it provides a way of replicating DNA. In order to replicate its DNA, a cell first divides the 2 strands, separates the 2 strands, and then wherever there's a C on one strand, it puts a G. Wherever there's a T, it puts an A. Wherever there's an A, it puts a T, and so forth. You do the same on the other strand, and you end up with 2 DNA molecules that are the same as the original DNA molecule. This happens every time a cell divides. Before it can divide, it has to replicate all of its DNA. The genome refers to an organism's complete complement of DNA, all of the DNA sequences in the organism. And of course, the human genome isn't just 7 base pairs. In fact, it's greater than 6 billion base pairs long. So inside every one of your cells, you have more than 6 billion pairs of G's, C's, A's, and T's. This is divided between 46 chromosomes. You get 23 chromosomes from each parent, and then there's a little bit of DNA in your mitochondria. And if you were to unravel all of the DNA from your chromosomes, it would be very long. Specifically, if you took the DNA from a single cell and unwound it, it would be about 2 meters long. So you've got this much DNA in every single one of your cells. And if you consider that the human body has, by a conservative estimate, about 10 trillion cells, then this means that you have 12 billion miles of DNA in your body if you were to unwind it all. And to give you some sense of perspective, that's 130 times the distance from the Earth to the Sun. Now, this seems ridiculous. This doesn't seem believable. The reason that this is possible is because DNA is also very thin. It's 2 nanometers wide, that 2 billionths of a meter, or about 1/50,000th the width of a human hair. So you're able to compress these really long strands of DNA into each of your cells. You can also think about how much DNA we have in terms of information content. So 6 billion base pairs, where you can have 4 different possibilities at each position, corresponds to 1.5 gigabytes of information storage capacity per cell. And again, if you multiply that by the 10 trillion cells in your body, that means that you have about 300,000 times more data stored in DNA in your body than in the entire internet. Granted, it's the same information repeated over and over, a little bit like this. So what do you do with all that information? What's that information used for? One of the most important pieces of information you have in your DNA is the information that's used to make proteins, a different type of biological molecule. I should mention for RNA fans in the audience, and I know there's at least one RNA fan in the audience, that this occurs by means of an intermediate called RNA, which is chemically similar to DNA with a couple of minor chemical differences. So I've got to, another piece of terminology that I'm going to use is a gene. There are different ways of defining what a gene is, but for our purposes tonight, I'm just going to define a gene as a segment of DNA that contains the information needed to make a protein. Now I've got to clarify something about proteins. When I say proteins to most people, this is what they think of. They think of foods that contain proteins. And that's not exactly what I mean. What I mean by protein is something a little bit more technical. It's a string of amino acids. It's a molecule, a long molecule that's made of a string of building blocks called amino acids. So just like DNA is made of a string of DNA bases, a protein is made of a string of amino acids. And like the DNA bases, amino acids are abbreviated with a bunch of letters. And there are 20 different types of amino acids that are found in proteins. The amazing thing about these strings of amino acids is that they're not just floppy strings, they actually fold into a well-defined three-dimensional structure. So they're kind of like self-folding functional molecular origami. The cell produces this string of amino acids and it spontaneously folds itself up into a three-dimensional structure that plays some functional role. So there are lots of different proteins. There are the motor proteins that move your muscles. There's hemoglobin that transports oxygen. There are many examples that we're going to encounter throughout this talk. So you might be scratching your head and asking yourself, so he said that there are 4 different DNA bases, but there are 20 different amino acids. So how is it possible that 4 DNA bases can encode 20 different amino acids? And the answer is that each amino acid is encoded not just by 1 base of DNA, but by 3 bases. So the genetic code is the rule that links triplets of DNA bases to amino acids. So for instance, AAG encodes an amino acid called lysine, which is abbreviated for some reason by K. And again, this happens by way of an RNA intermediate. Another thing that I'd like to clarify about proteins is that, so we've all heard that you need proteins in your diet, and that's true, but you don't need the proteins per se, you need the amino acids in the proteins. So when you eat food containing proteins, those proteins are broken down into their constituent amino acids, and then your cells reassemble these amino acids into human proteins. So it's important to note that the proteins in your diet are not the same as the proteins in your cells. The proteins in your cells are made from the amino acids that are derived from the proteins in your diet. Okay, so let's summarize so far. I've shown you that DNA is an information storage device that uses 4 chemical letters, A, T, G, and C, instead of zeros and ones. And it has an enormous capacity. It can store 1.5 gigabytes per cell. And part of that information is the instructions for making proteins. So this seems like a great system. There's a problem, though, with storing information in a molecule, and that's that molecules can undergo chemical reactions. So the information in DNA can be damaged. And this is a serious problem that the cell faces, and it's something that evolution has devised a lot of solutions to. So let's talk about different types of DNA damage. Many of you have heard of this dangerous molecule that's found in a lot of different places. It's found in each one of your cells. In tumors, overconsumption of it can kill you. It's known as dihydrogen monoxide, also known as H2O or water. And in fact, water can react with DNA. It reacts very slowly. You've got a 1 in a million chance at each base of it reacting with water. But when DNA reacts with water, it can cause the loss of individual bases. And like I said, this is a very slow reaction. It doesn't happen very often for any given base, but since you have 6 billion bases per cell, this actually happens thousands of times per cell per day. And if you didn't fix this, this would be a disaster. Another example, let's say that you're out at the beach, not wearing sunscreen, not a good idea. So your DNA is being exposed to ultraviolet light from the sun, very energetic light from the sun. And that can cause adjacent bases in the DNA to get fused together chemically. And this DNA then becomes unreadable. This is also a disaster. Fortunately, we have systems inside our cells that fix these types of damage and various other types of damage. So there are proteins in the cell that come by and clean up the damage and then fill in any missing information. And in these cases, those proteins take advantage of the fact that the information in DNA is stored in duplicate. So even if one strand is damaged, the other strand can be used as a backup copy to provide the lost information. It can be recopied onto the damaged strand. But what if both strands are damaged? And this is what happens in the type of damage that I study called the DNA double-strand break. So a double-strand break, as the name would imply, is when both strands of DNA are sheared apart. So this is a very serious problem. The cell somehow needs to put its DNA back together. And so first I'm going to talk about different ways that double-strand breaks can arise. One way is due to radiation. And this may seem really exotic, but in fact we're all surrounded by radiation all the time. From cosmic rays, from minerals in the soil, from potassium-40 in our own bodies. And so I don't want you to freak out and go build a lead bunker because, in fact, the cell has ways of dealing with this. But first, let me describe what happens. So let's say a gamma ray passes through your cell. This can strip electrons off of water molecules through the path of near the path of the gamma ray, and it produces this cloud of reactive molecules, including hydrogen peroxide and free radicals, which you might have heard about last time if you came to the last Science in the News lecture. And then these reactive molecules can react with DNA, and in some cases cause both sides of the DNA, both strands, to be chemically broken. Another example of a way that a double-strand break can occur is as a consequence of other repair processes. So let's say that this DNA molecule reacts with 2 different water molecules, and it loses 2 bases on opposite sides. Now when the cell tries to repair this damage, it will inadvertently generate a double-strand break. And these are just 2 of the ways that double-strand breaks can be generated. There are other ways as well. But whatever their source, it's estimated that there are somewhere on the order of tens of double-strand breaks that occur in each of our cells per day. Yet a single unrepaired double-strand break can kill a cell. So fortunately for us, since you all still seem to be alive, we have DNA repair mechanisms that can repair double-strand breaks. The one that I study is referred to as end joining. And so in end joining, a bunch of proteins, which I'm going to talk about in more detail later, bring the 2 DNA ends together, they fill in any gaps, and then they chemically join or ligate the 2 strands of DNA together. Very easy, right? Unfortunately, there are a couple of different types of errors that end joining can make. And in this case, an error will lead to a permanent change in the DNA sequence, which is known as a mutation. So let's say that instead of lining up the DNA ends like this, the end joining machinery lines them up like this. They're still base paired properly, but now you have a couple of bases kind of dangling off the end. Well, there are enzymes in the cell that can clip off these bases and then rejoin the ends, and you fix the break, right? I mean, this is still repair of a double-strand break, but unfortunately, you've lost 2 bases, 2 base pairs. So this is one problem with end joining. This is one difficulty. The other difficulty happens when there are 2 double-strand breaks in the same cell. So let's say that we have 2 chromosomes, DNA piece 1 and DNA piece 2, and double-strand breaks occur in both DNAs. Now, the correct way to repair this would be to take these 2 ends and put them together, and take these 2 ends and put them together. But occasionally, the end joining machinery performs a switcheroo and joins this end to this end, and this end to this end, and generates what's known as a chromosomal translocation. Chromosomal translocations are very frequently found in cancer, and some of them occur so often or have such grave consequences that they have special names. And a famous example is the Philadelphia chromosome, which arises when a double-strand break occurs near the ABL gene on chromosome 9 and near the BCR gene on chromosome 22. And then if these 2 double-strand breaks are incorrectly joined, if you join a piece of chromosome 22 to chromosome 9 and vice versa, then this brings together the 2 genes, BCR and ABL, to make what's called a BCR-ABL fusion gene. And this produces a new protein that the cell has never seen before. It's called the BCR-ABL fusion protein. And this protein, this fusion protein, directs cells to grow uncontrollably and can cause different kinds of leukemia. I should note as an aside that it's really useful to know this because different companies have been able to develop inhibitors of this BCR-ABL fusion protein, and that's dramatically improved the prognosis of patients whose cancers involve this translocation. So it's good to know the molecular details of what causes genetic diseases like cancer. So in the long run, I'm interested in trying to understand how does the end joining machinery avoid making terrible errors like this? How does it avoid unnecessarily deleting genetic information? And how does it avoid putting the wrong chromosome pieces together? But in order to answer that question, we first need to answer a more basic question, which is how are the 2 DNA ends held together in the first place? I've drawn the end joining proteins as kind of this diffuse blue circle, but we'd like to fill in the details of that blue circle and figure out exactly what's holding the DNA ends together. And so stay tuned to the next part of my talk where I'll discuss that in more detail. To summarize part 1, DNA is an information storage device. That information is stored and a sequence of chemical letters, A, T, C, and G. That some of the information in DNA is used to make proteins, and a protein is a string of amino acids that folds itself into a unique three-dimensional structure and performs some function. DNA can get damaged in various ways, and there are various repair processes that fix this DNA damage. Without these, we wouldn't last very long. Unfortunately, DNA repair, like everything, can make errors, and errors in DNA repair lead to mutations, which are the root cause of genetic diseases, including cancer. So that ends the first part, and do you have any questions? Yes?
You said that there were tens of, your cells would see tens of errors.
Mm-hmm.
And are you mean that every single cell would see 10 per day, or your body would see 10 per day?
Every single cell. So that estimate was made by taking cells and exposing them to increasing doses of radiation and seeing how many, so it's known how many double-strand breaks a given dose of radiation will produce. You can do an experiment with DNA in a tube and see how many breaks are produced. And so they look at, they stain the cells in such a way that it detects double-strand breaks, not with 100% efficiency. So you can compare the number of spots that you see in response to increasing doses of radiation with how many spots there are normally in the absence of radiation. And from that, you can kind of estimate how many double-strand breaks occur during a normal cell division cycle. It's not a perfect estimate. I think the number that might not be a totally accurate number for the entire body. It's somewhat difficult to estimate. Yeah. Yeah.
Repeat the question.
Oh yeah. Yeah. So that question was how are the 10 to 50 double-strand breaks per cell or across the entire body? That's about the right order of magnitude for a single cell. Yeah. Any more questions for this part? Perfectly lucid. Yes.
So now, not every cell in the body reproduces at the same rate.
Right.
So therefore, these 10 to 50 errors will affect some processes in your body more than others.
Yeah. Yeah. So dividing cells in general have more DNA damage or are more susceptible to DNA damage than undividing cells. That's actually the basis for cancer chemotherapy, or that's one way that cancer chemotherapy probably works, by selectively targeting rapidly dividing cells, which are more susceptible to DNA damage. Yeah. Yeah.
Is DNA ever damaged to the point where it just becomes useless?
Yeah, so if the DNA in a cell is damaged beyond repair, if the cell doesn't fix the damage within a certain period, then the cell actually undergoes a type of programmed cell death called apoptosis. It commits suicide. And the reason that you'd want to have a mechanism to do that is because if the DNA is so horribly damaged that it can't be rapidly repaired, then there's a good chance that if you tried to repair it, you would end up with these chromosome translocations, and that might cause cancer. So rather than putting the rest of the organism at risk, that cell is programmed to commit cell suicide. Yeah. Yeah.
You say that evolution has repaired some mutations over time.
So could you repeat the question? I'm sorry.
Did you say that evolution has repaired mutations in humans over time?
Yeah, so the question is whether evolution has repaired mutations in humans over time. So the repair processes that I'm talking about occur within cells. So the cell has mechanisms for repairing its DNA when it senses that it's damaged. And so we're going to talk in the end about how mutations can sometimes be useful, but actually one situation in which mutations can be useful is for evolution. It's mutations that ultimately provide the genetic diversity that natural selection acts on to cause changes in species. So yeah, mutations are kind of a double-edged sword. If you have too many of them, then it can be detrimental. You can destroy the information that you need to make proteins and do other things. But if you had no mutations at all, then there would be no evolution. Yeah.
Yeah, that's kind of my question with the translocation and fusion of the proteins. It doesn't always have to be dysfunction.
Yeah. So yeah, some mutations can be completely benign, and some mutations might be beneficial. Probably most mutations in the sequence that encodes a protein will either be neutral or detrimental. Only a small fraction of them will be actually useful, but it's that fraction that is acted upon by evolution to increase the frequency of that mutant gene in the population. Yeah. I think we're going to take a very short intermission, and then we'll go to parts 2 and 3. Yeah. Thanks a lot. If you have any more questions, I'd be happy to answer them. Thanks. Yeah, so one problem is that often machinery that's designed or that is evolved to destroy damaged cells is inactive again. So that's one of the things that, so yeah, so you might have heard of the gene. Okay, so there's a gene that was a protein called E53. And that's one of the things that senses cell state breaks. And if they hang around long enough, then it can help the cell kill itself. So E53 is very often mutated in cancer. It's one of the most commonly mutated proteins in cancer. And that's because if you mutate P53, then you can be as damaged as you want it and still keep growing. Yeah. So cancer is really sneaky. Yeah. Yeah. And people are trying to one goal in chemotherapy is to reactivate this ability of cells. Yeah. Cancer cells, I mean, it seems like cancer cells are living anyway. They're subjected to all of this. They're subjected to increased levels of damage of cell replication structure. And so they're kind of poised on a knife edge. They could kill themselves. So normally there's a whole network of proteins that signal to each other that control the rate of cell there's this cell cycle. So yeah, and there are a bunch of proteins that regulate that. There are proteins called cyclins that go up and down the cell cycle. Cells go up and down, and then there are proteins called kinases that attach phosphate groups to other proteins, and you can transmit cells that way. Their activity depends on the cells being sequence. So there's really this big network of all these proteins that are communicating with each other. Is not pure on. You got all these big network of proteins that communicate with each other, decide whether the cells can divide or not. And if you have a mutant protein like BCR-ABL, it starts sending signals rather than sending the signals in the other way. It sends signals. Yeah. So something that I didn't really explain is that DNA has polarity. So there's it's opposite. So the two ends for So if you were to take this and flip it over, you would have that. So that yeah. So that's really this question. Yeah. So. So this. So this end, it has a phosphate group. So the backbone, I kind of simplify things a little bit. We call these all bases. This is where I am base. So you've got on the backbone, it's phosphate part of phosphate. And so this is the polyprimate. So you've got phosphate sugar, phosphate here, phosphate sugar. This is the phosphate, So yeah, if you were to flip it, yeah. So if you were to flip it over, so this one, 5' to 3' this way. This one, 5' to 3' that way. Yeah. So there's no way you have to rotate it. You have to rotate it like this. Yeah. That's gonna fix it. Yeah.
Okay, so do that. Great.
It's so I mean there are situations I'm going to talk a little bit about it, but there are situations where you can have an overhang on one strand and it can actually fold back over. Yeah. And form something that's called a hairpin. So let's say that you had let's say that you have an overhang that's partially, this one's partially covering itself, so it can fold back over the hairpin in case it's plugged in together. And then this is kind of difficult to do it, so it's much better than that. Yeah. And actually, we're going to talk about antibody sequencing, but antibody sequencing is often so this will make sense of that, but antibody sequencing is often repeats at seconds. That's because they're produced by yeah.
You should like practice. We should have that. Remember one we're I'm going to one more time for a minute. What? We have one ready to go to. We have one where he sleeps on the also some contact.
So we have two strands of DNA. So I mentioned that proteins are made. You've got DNA, and then you have RNA, and then you have protein. So the way that RNA is used is you actually open up a hole in the DNA, and then there's a protein called RNA polymerase that makes the DNA, or makes the RNA, this is the RNA, based on this segment of DNA inside the molecule. So the RNA always has the same sequence as this DNA strand. And the directionality there is enforced by a sequence of DNA called the promoter. Yeah. No. So you can have one gene, usually represented as the best gene. You can have one gene right here, and then the next gene right here. So in this gene, the top strand, the RNA that's next to the top strand of the DNA will have the same sequence. And this gene, the RNA will have the same sequence as the bottom strand of the DNA. So it's important. Yeah, it goes. Yeah, sorry. This is kind of technical way around this. Yeah, so you can have for one gene, the RNA is being produced with this DNA, this strand of DNA as a template, so it has the same sequence as this strand of DNA. And then over here, you can do the opposite thing. Have the RNA produced with this strand as a template, so it has the same sequence as this strand. Yeah, basically, yeah. Yeah, so I mean, like, would it ever like? So normally proteins are only produced using RNA that's made in a specific direction. There are situations where you can have a region of DNA that's satisfied in both directions, or that's RNA is made in both directions, but that's kind of a special case. It's often associated with turning adjacent genes off. But yeah, normally for producing proteins, it's always yeah, that's not usually the case. But that could happen. So actually, humans, this is another complication. Humans, the gene doesn't look like this, but it actually looks. You have a lot of segments, exons, and then intervening segments on the fronts. And when you make an RNA from this, all of the exons are spliced together. I don't know if that makes sense. So so you take out the exons. So sometimes you can have another gene coming in the opposite direction that has. I think this is not very common. That has exons like that. So now this exon doesn't mean anything. It's perfectly in front of this gene. Yeah. This is why I made a disclaimer when I defined gene, because human cells especially, it can become unclear exactly what a gene is. You have all these different exons and introns, and genes can overlap, and you have this promoter sequence that controls the directionality of producing RNA. And then to top it all off, you have these sequences called enhancers that tell the promoter what to do. So I just washed my hands with that and said that when it goes to protein as a gene. Yeah, sure, of course. Yeah, so the DNA is made of 4 layers. RNA is made of 4. It also has B, C, and G. It has another weird letter U that makes it S and T. There's some slight differences. And then so this step of going from RNA to protein happens on a machine that's called ribosome. So the ribosome looks kind of like that. It scans along the RNA, and then it erases this string of amino acids based on that.
Thank you. I think we'll get started again.
Okay, everyone, thank you for sticking around. So I ended the last section on a cliffhanger with the question that I'm interested in answering in the lab, which is during end joining of broken double-strand DNA ends, how are the ends held together? This is a question that I've been trying to address for the last couple of years. Fortunately, we knew from work from previous groups the identities of a lot of the proteins that are involved in this process of end joining. But what we didn't know was how all of these proteins interact with each other and with the DNA ends to hold the ends together. And we didn't even know for sure which proteins were involved in the formation of this blue circle, or this complex as we call it. You don't have to know the names of most of these proteins, fortunately for you, because they're all kind of weird acronyms. But I'm going to mention 2 of them. One that I'm going to talk about more called XLF. And then one that was just discovered called PAX. The discovery of this was reported just about a month ago. So it's possible that there are components of this blue blob that we still don't know about. Now, end joining happens inside cells normally, but it's kind of difficult to study all these nitty-gritty biochemical details in cells. And so what we like to do, what people figured out that you could do a bit over a decade ago, is take cells and mash them up, take the soluble portion, and make an extract that contains all of the proteins that were originally in the cell. And amazingly, this extract, which contains all the end joining factors, if you add DNA fragments to it, can join them together. And this depends on all of the known end joining factors. Now you can do this with different types of cells. For convenience, we used a kind of odd type of cell, which is frog egg. So the reason we use this is because you can obtain frog eggs in large quantities, and you can make lots and lots of extract from them. And also, they perform this end joining reaction at room temperature. So we can do everything at room temperature. We don't have to heat it up to 37 degrees Celsius like the human body. Now, let's say that you were a skeptical member of Congress and said, oh, why are you spending our money studying end joining in frogs? Well, the answer to that is because we and frogs are descended from a common evolutionary ancestor. The end joining process in frog cells is very similar to the end joining process in our cells. And so we can learn about the basic machinery of DNA end joining by studying cells from any vertebrate, including these frog eggs. My goal in this project was to develop a way of watching individual pairs of DNA ends getting joined together using a microscope. And this sounds like a totally quixotic goal, but in fact it's not. We can look at individual DNA molecules under a microscope, and here's an example. So these are individual 5,000 base pair long DNA molecules. They've been labeled with a fluorescent dye, tethered at one end to a glass surface, and then extended by flowing a solution past them that contains this fluorescent dye. So I need to explain a bit about exactly how we do this. So first of all, we build all of our own microscopes, and so we give them cool names. This one is named Chuck Norris. I like to say that I don't do experiments on Chuck Norris. Chuck Norris does experiments on me. We use a few different tricks to be able to look at individual molecules. The first trick I alluded to before is something called fluorescence. Basically, fluorescence is when you shine light of one color or one wavelength on a molecule. It absorbs that light, and then it re-emits light of a different color. And we can separate these two colors of light using colored filters and distinguish the light that went in from the light that comes out. Another thing that we do is we attach the molecules to a glass surface, and we illuminate them with a laser at an oblique angle so that we just illuminate a very thin slice of the sample. And that way we can look at everything in this thin slice, but not get background from the rest of the sample. And another thing that we do is we use a very sensitive camera. So it's kind of like the CCD camera that's in your, I'm not gonna pull it out, I'll mess up my microphone. It's kind of like the camera that's in your phone. It's the same basic principle. It's a charge coupled device, but this has some additional layers to it to amplify the signal. And it's also cooled to minus 70 degrees Celsius to reduce background noise. So we use this very sensitive camera that can detect light from individual molecules. So I don't just want to look at a single molecule. I want to look at the two ends of a piece of DNA. I'm not interested in just looking at the DNA. I'm interested in looking at the relative position of two DNA ends. And the way that I do this is using a phenomenon called forced to resonance energy transfer, or FRET, which you can think of as a molecular ruler. And the way that this works is that when one fluorescent molecule, we're going to call this the green fluorescent molecule, gets close to a second fluorescent molecule, I'll call it the pink fluorescent molecule, a fraction of the time, it can transfer the energy that you put into it to this pink fluorescent molecule. And so now instead of just seeing fluorescent emission from the green molecule, you also see fluorescent emission from the pink molecule. And again, we can distinguish these different colors of light using different colored filters. Something that I should mention is that this blue light is only absorbed by the green molecule. So it's not that we're aiming the blue light at the green molecule. We're illuminating the entire sample with this blue light, but it's only absorbed by the green fluorescent molecule, but it's not absorbed by the pink fluorescent molecule. The important feature of FRET is that it depends on the distance between the two fluorescent dye molecules. If the two fluorescent molecules are very close together, then the green molecule transfers the energy that you put into it to the pink molecule virtually 100% of the time. And if they're very far apart, the green molecule practically never transfers the energy that you put into it to the pink molecule. And then if they're at some intermediate distance, there's a 50% probability that the green molecule will transfer its energy to the pink molecule. And this all happens very fast. This happens on the scale of nanoseconds. And so basically we're looking at the average of what fraction of excitations of the green molecule gave green light versus pink light. So we're just looking at intensity from the green molecule and intensity from the pink molecule. When they're close together, it's more pink than green. When they're far apart, it's more green than pink. And when they're in the middle, then green and pink are about equal. Okay, so how did I apply this to look at joining of DNA ends? Oh, another thing I should mention is that the halfway point here is 5.6 nanometers, which, if you'll remember, the width of a piece of DNA is about 2 nanometers. So this is about the perfect length scale to be able to look at molecular sized interactions. Okay, so this is how I applied this to look at the interactions between DNA ends. I made a piece of DNA that's 2,000 base pairs long, that's labeled at one end with a green fluorescent dye, and at the other end with this pink fluorescent dye. When the two ends are not tethered together, they're flopping around, and on average, they're not very close together. And so there's a low Fred efficiency. You see a lot of signal from the green fluorescent dye, but not a lot from the pink dye. If I add frog egg extract and proteins in the extract bring the two ends together, this end joining complex bridges the two DNA ends, then I can start seeing energy being transferred from the green dye to the pink dye. And so I see both green and pink fluorescence. And here's an example of what actual data look like. So here I'm plotting the fluorescence intensity from a single fluorescent spot on the surface of the glass cover slip. At the beginning of the experiment, before I add egg extract, the two ends are flopping around randomly, and I see green fluorescence, but not pink fluorescence. But then, over time, at some point, after adding the egg extract, the two ends are brought together, and I see a dip in green fluorescence and an increase in pink fluorescence, indicating that energy is being transferred from the green fluorescent dye to the pink fluorescent dye. And I'm saying this is also known as FRET, and that means that the two ends are close together. I can look at multiple back and forth transitions. I can monitor a single pair of DNA ends and watch them come together and fall apart. So here's an example of some of those data. The two ends start out apart, they come together, they fall apart, they come together, they fall apart, they come together, and they stay together. This is a plot of the Fred efficiency. This is a little bit noisy, but you can see they come together, they fall apart, come together, they fall apart, and they come together. And I can look at this not just on an individual molecule, but on hundreds of molecules per field of view. And I can look at how the fraction of molecules that are bridged increases over time. So this is the fraction of molecules that are pink divided by the total number of molecules that are either pink or green. And I can see the DNA ends being bridged over time after adding the egg extract. So my original problem was I wanted to figure out what's responsible for bridging the DNA ends. And the way that we test this in egg extract is by taking away proteins one at a time. In order to take away a protein specifically, you need something that binds to it specifically. And in our case, we made antibodies. And so the way that you make an antibody that's specific to a particular protein is you take a fragment of the antibody. This was all done by a company. So we sent the amino acid sequence of this XLF protein that we're interested in to a company. They chemically synthesized a small fragment of the XLF protein and injected it into rabbits. And then the immune systems of the rabbits raised antibody molecules that bound XLF specifically. And so now what I could do is I could take these antibodies that were derived from the rabbit, attach them to microscopic beads, And what this serves as is a sponge for XLF. It's kind of like an XLF sponge. It binds my protein of interest specifically in the extract and takes it out of the extract. So if I do a couple of rounds of this, it's called immunodepletion, I end up with extract minus XLF. Everything else in principle is still there. The only thing that's been removed is XLF. And if I test that XLF depleted extract in my experiment, I find that it's no longer able to bridge DNA ends. It's completely dead for DNA end bridging. Now, you might object to this and say, well, what if the antibody bound to a different protein as well, another protein that's important for bridging the DNA ends? And so to exclude that possibility, we purified XLF protein. I won't tell you the details, but we made it in bacteria and then purified it and added that purified protein back to the extract. And we found that that completely rescued the effect of the XLF depletion. So the only difference between this red curve and this black curve is the presence or absence of this purified XLF protein. And so we can tell from these data that XLF is required to bring DNA ends together. It's a part of this complex, this end joining complex, that tethers DNA ends together so that they can be rejoined. And I did these experiments for a variety of different proteins, and I did a slightly different setup of this experiment that I don't have time to go through. And the end result, what we conclude from all of these data, is that there are actually two stages of end bridging. So where we had one stage before to worry about, now we have two stages to worry about. There's a stage where the two DNA ends are tethered together, but they're not very close to each other, so we can't actually see a fret signal between the two DNA ends. And then there's a stage where they're right next to each other, and so we can see the fret signal that I was talking about. And we know that a couple of proteins are involved in the formation of this long-range complex. If you can think of a more creative name, I'd be all ears. And then XLF and a couple of other things are needed to transition from the long-range complex to the short-range complex. So as is often the case in science, we've answered one question partially, and it's led to a lot of new questions. What on earth are these two different stages? Why does the cell go through the trouble of doing this? I think it might be some sort of error control mechanism. That's speculative. And another question that we're interested in is, where does processing of the DNA ends occur? So I showed you at the beginning, sometimes the ends need to be processed. You need to fill in missing bases and stuff in order to join them back together. And does that happen in the long-range complex? Does it happen in the short-range complex? Does it happen before they even come together? We don't know yet. So that's something that we're interested in investigating in the future. So to summarize this part, the goal of my work has been to determine what holds DNA ends together during end joining repair, which proteins are involved in particular. The approach that I took was to use single molecule FRET imaging and some other single molecule techniques that I didn't have time to discuss. And I combined that with removal of proteins from the extract with antibodies to see which proteins are necessary for different stages in this bridging process. And from these experiments, I was able to conclude that there are two distinct steps in the bridging of DNA ends. We still don't know exactly why that is, and they depend on different proteins. So now we're working to try to figure out, why are there two stages? How do you go from one stage to the other? And what's exactly happening in each stage? So do you have any questions about this part? Yep.
This is sort of a process question, but would a mock depletion using a different antibody have been as good of a control as the re-adding in the XLF?
So actually, so the question was, would a mock depletion with an unrelated antibody be as good as adding back XLF? And so what I didn't mention is that by not XLF depleted, I meant mock depleted. So this was actually, we thought of that. We depleted this with a completely unrelated antibody. We took a completely unrelated antibody that shouldn't deplete anything relevant, bound that to beads, and did a mock procedure where we did the same incubation of the extract with beads, and that's still able to join the DNA ends. But we wanted to be totally certain, and so we added back the purified protein.
How long, on average, does it take to do this joining of a great DNA?
Yeah, so in cells, I guess we don't have we don't have a great way of measuring how long it takes in cells because you can't, it's difficult to look directly at whether the ends are joined or not in a living cell. This is using, this is putting a sample on a cover slip and using fluorescence, single molecule fluorescence imaging to see if the ends are together. It's estimated that it takes somewhere on the order of hours, a couple of hours for end joining. It takes slightly longer for a different double-strand break repair mechanism that I'm going to mention briefly at the end. But I think that this is the most direct way that this and also the bulk experiments and extract of actually observing how long this process takes. But whether this is really representative, if the timescales here are representative of what's happening in cells, that's a different question. It might happen slower or faster in an intact cell. Yeah. Great. Well, if there are no further questions on this part, I'll go on. And in the final part, I'd like to talk about how changes to DNA or mutations might be useful in some cases. So I talked about mutations mostly like they're really bad things that you want to avoid, but in some cases, you actually want to cause changes in DNA. And I'll talk about two cases like this. One that's happening in your bodies right now, that it is required to make antibodies, a diverse range of antibodies. And then a second technique that's recently become really prominent that molecular biologists have figured out how to do called gene editing. So we talked, I mentioned antibodies briefly before. An antibody is a molecule that binds specifically to some other molecule. And antibodies are useful, are produced by your immune system to target germs, to target things like nasty bacteria. So let's say a nasty bacterium invades your body. Antibodies are produced by a type of immune cells called B cells. Each different B cell makes a different type of antibody. More on that later. And because you make so many different antibodies in your different B cells, there's a good chance that at least one B cell will be able to specifically bind to a molecule on the surface of this nasty bacterium. So if the B cell is able to bind to the surface of this bacterium, it signals the B cell to start growing and dividing and making lots of copies of itself that make the same antibody. And these B cells secrete the antibody into your bloodstream, and this unfortunate bacterium is eliminated by the immune system, is targeted for destruction by the immune system. This begs the question, though, how do you make so many different antibodies? How is it that different B cells make different antibodies, given that they have the same genome? Well, the answer is that they actually don't have the same genome, because they rearrange the genes that are involved in making antibodies. So the genes that you inherit from your mom and dad, the antibody genes that you inherit from your mom and dad, are kind of like furniture from IKEA. It doesn't come pre-assembled, you have to assemble it. And so this happens in developing B cells. What happens in a developing B cell is that there's a protein that cuts between these different segments in the antibody gene. It always cuts between the segments, but which segment it cuts next to can vary. And then the intervening sequence gets lost, and the two ends are joined together by end joining. So technically, this is a mutation. It's a change in the DNA, and it happens a couple of times, and the ends are resealed by end joining. The advantage of this is that you can make a whole library of different antibodies. So by cutting next to different segments and joining different segments, you can make a variety of different combinations of these segments. And I've only shown 2 of each segment here, but in reality, you have many more than that. And so you can make millions of possible combinations of antibodies. It's actually even more than that, because the sloppiness of end joining adds some additional diversity at the junctions between these segments. And with all these combinations, it's likely that your body can find an antibody to recognize any foreign virus or bacterium. So we're not able to do anything quite this elaborate yet as molecular biologists, but we are able to exploit a similar trick of cutting DNA in order to make targeted changes. So the overall idea is we take a protein that can cut DNA in a specific location, that double-strand break, when it's repaired, will sometimes generate a mutation. And so you can target mutations at the specific gene you're interested in using a protein that cuts at that specific site. And there are a couple of different proteins that do this that have been developed over the years. They're called things like zinc finger nucleases and talons. But something that's really revolutionized this field in the last couple of years is a new enzyme that's known as Cas9. Some of you might have heard of it. It goes by another name, which is CRISPR-Cas9. You might have heard of CRISPR or Cas9. It's the same system. Cas9 was, the way that Cas9 works was figured out by several different groups, but one of those groups came from a very unusual place. They came from Danisco, a yogurt company. They make cultures for yogurt. And it turns out that a big problem in yogurt manufacturing is that the bacteria that make yogurt are often infected by viruses called bacteriophages. And so you can destroy an entire yogurt culture if it's infected by viruses that attack these bacteria. And Cas9 is part of a bacterial immune system that cuts the DNA of the invading viruses and thwarts the viral infection. The useful property of Cas9, however, is that it doesn't just cut DNA from viruses. The way that it's targeted in a specific DNA sequence is with a short piece of RNA, this DNA-like molecule, called a guide RNA. So you can take Cas9 and mix it with a guide RNA in a tube and get it to target whatever DNA sequence you want. That's a really powerful tool. And unlike previous DNA cutting proteins like zinc fingers and talons, you don't have to re-engineer the whole thing every time you want to cut a DNA sequence. You can use a single protein, mix it with different RNAs, and get it to cut DNA sequences. And so here's how this would work. Let's say that there's a gene that you want to interact, or that you want to interrupt or disrupt in whatever cell type you're growing. You can buy Cas9 commercially and order a custom RNA online, have it FedEx to you, and then you can introduce these two things into your cells. They will produce a targeted incision in the DNA, generating a double-strand break. And then that might be repaired correctly, but if it's repaired correctly, the enzyme just cuts again, the protein just cuts again, keeps doing that until eventually you get a mutation that disrupts this gene that you're targeting. So this is a great way of targeting a specific gene. And it's been employed in a lot of different organisms. This is just over the last couple of years. It's been employed in a lot of different organisms. Any organism with a DNA repair mechanism can be targeted for gene editing using Cas9. And so looking at this list, looking at rabbits, mice, monkeys even, you might ask, could you do this in humans? And actually, from a technical perspective, there's no reason why you couldn't. It works in human cells in a dish. You could probably make genetically modified humans using this Cas9 trick. Of course, you might not want to. There are a lot of ethical issues with this. And in fact, in the last week, there was an editorial published in Science magazine that argues that we should place a moratorium on this. I'm kind of in favor of that myself. And there's an article that's going to be coming out in Nature, in the print version of Nature tomorrow, that's arguing kind of the same thing. You can also read about it in the New York Times article that I mentioned on your handout. Now, of course, this is really super controversial, but fortunately, there are less controversial ways of using gene editing for therapeutic purposes. And one idea is that it could be used for gene therapy. So let's say that we have a patient with a disease. You could harvest cells from this patient, edit them using Cas9 plus a guide RNA to cut a specific gene. Let's say that there's a specific gene that you could inactivate and it would improve the function of these cells and allow you to treat a disease. And then you can introduce the cells back into the patient and potentially cure the disease. So I'll give you one example of how this is currently being tried, something that's currently in clinical trials for treating HIV. So as you may know, HIV infects immune cells called T cells. And in order to bind to a T cell, the most common type of HIV has to bind to a protein on the surface of the T cell called CCR5. In patients who don't have CCR5, those individuals, there are some people who have a naturally occurring mutation in CCR5 that makes them resistant to infection by HIV. So the idea is, if we could remove CCR5, we might be able to make T cells resistant to infection by HIV. So here's the idea. You harvest T cells from the patient that have a normal CCR5 gene, and if they're cut by a DNA cutting protein, like a zinc finger nuclease or Cas9 plus a guide RNA, this generates a double strand break, and then occasionally that will be rejoined, generating a mutation that eliminates CCR5 protein and makes these cells impervious to infection by HIV. This is currently in phase 2 clinical trials from a company called Sangamo Biosciences, and they happen to be using one of these older approaches involving a zinc finger nuclease rather than Cas9. So the phase 1 results from this were released in 2014, and that wasn't sufficient to show efficacy, but it looked promising nonetheless. So only time will tell if this is actually a viable way of curing HIV, but it's a very exciting development. One thing that I'd like to mention before I end is that Cas9 can be used not just to generate random mutations in a gene, but it can also be used with the help of a second DNA repair mechanism that I didn't have time to discuss called homologous recombination to introduce specific new sequences into a target site. So let's say that you inherited a gene mutation that inactivates the function of some gene, and you want to restore it to the correctly functioning gene. So the idea is that your doctor could introduce Cas9 plus the guide RNA into these cells, along with a short segment of DNA called the donor sequence, and then using the second DNA repair pathway, homologous recombination, this donor sequence could be introduced or recombined into this target gene, producing a now functional version of the gene. So this is still pretty hard to do. It's a lot harder to do this than the sloppy, imprecise mutation making with end joining, because we still don't know how to prevent cells from doing end joining and instead do homologous recombination. But that's kind of the holy grail of the field, to be able to make these precise edits in a genome using this technique. So to summarize part 3, edits in DNA can sometimes be useful. They can be useful both for making antibodies in your body or for editing genes for researchers. So I already mentioned that. In order to make targeted mutations, we can cut DNA with programmed DNA cutting proteins. And one of these is Cas9. That's one that's become very prominent in the last couple of years. And this sort of gene editing approach could be useful both to study functions of different genes by inactivating them, or possibly for gene therapy by altering disease genes and returning them to a state of normalcy. With that, I'd like to personally acknowledge Science in the News for giving me the chance to give this talk, and in particular Steph and Jess for organizing this lecture series. I'd like to thank Joe Laparo and Johannes Walter, my advisors. I'm co-advised by 2 professors at HMS, and my lab mates for making the lab a fun place to work. And I'd like to acknowledge our funding sources. We're funded by the Howard Hughes Medical Institute, by the National Institutes of Health, and by the National Science Foundation. And I'd like to thank all of you for your attention and your great questions, and also for paying taxes to fund these fine institutions. So thank you for your support of science. Thank you. If anyone has any more questions, yeah.
So what could be side effects from taking away the CCR5?
Yeah, so there are people with mutations in CCR5 already inactivating, where they have no CCR5 protein on their T cells, and there doesn't seem to be any ill effect of that, to my knowledge. So it's plausible that you could get rid of CCR5 in T cells, and they would still be functional T cells, but they'd just be resistant to infection by HIV. There might be some slight side effects, but I don't know that for sure. Yeah. Good question. Yeah.
Well,
Okay.
yeah, how long does the RNA guide have to be? So maybe
Right.
you show them the 75 size.
Oh, I can't remember the exact size. I mean, it's a short RNA. You can order it in a tube. and they just type in the sequence online, and then they send it to you in mail. So it's short enough that you can actually order it synthesized. Yeah, the region that binds to the DNA is just 20 bases. So and that's largely sufficient to guarantee specificity. The one problem with Cas9 is that it does have what are called off-target effects. So in addition to cutting at the site that you want to cut at, it can also occasionally cut at other sites. And people have been working to figure out how to reduce that. So that's one reason that trying to make a genome-edited baby right now would be really bad, because you might cause unintended damage, in addition to other reasons. Yeah. Yeah.
So you were talking about how your B cells are sort of rolling the dice and generating all these different combinations and hope that some of them will bind to some invader,
Yeah.
and then they'll multiply those. So, and you said there's maybe 2 million possible combinations. How many of those combinations are harmful to the human body itself, like autoimmune disease?
Yeah, so there's a second part of that I didn't mention, which is that your body weeds out B cells that are self-reactive. So there are mechanisms for identifying B cells that react to your own proteins, because that would be bad if you raised antibodies against your own proteins. So B cells may sometimes, I think I mentioned this in one of this program, cell death mechanism, where cells can destroy themselves if they're doing something harmful, and B cells can also do that. They can destroy themselves instead of producing this autoreactive antibody. That's a good question. Yeah.
So what happens if one of those cells, or some of them aren't used? Do they also just self-destruct? Or?
So unfortunately, I don't know. I'm not sure. I'm not an immunologist. But yeah, that's a good question. The question was if the B cells are not used, do they eventually auto-destruct? I think after a certain period of time, they stop growing, they stop replenishing themselves. But I'm not sure. I'm not an immunologist. Yeah. Thanks.
Maybe you don't know this one either, but is it known what all of those possible combinations would do, what they would attach to?
No, so it's an enormous range of possibilities. And actually the 2 million number that I quoted is a serious lowball estimate, because in addition to rearranging segments in different ways, you can also have different junction sequences between segments. So this end joining process can delete nucleotides. In this case, it can also add nucleotides, or it can add bases. The way that happens is there's an enzyme called terminal transferase, a protein called terminal transferase, that adds additional random bases at the broken DNA ends. So it adds just random scrambled sequence. And a lot of the time, that random sequence that's added at junctions makes the antibody completely non-functional. So a large proportion of antibodies are never functional to begin with. But that's the price that you pay for producing a very diverse range of different antibodies. Yeah. Yeah.
So are B cells inherently made to mutate versus other cells you want to repair them the way they were? Do B cells tend to want to diversify?
Yeah, so that's so the question was, do B cells want to diversify compared to other cells? So actually there are certain proteins that are made in B cells that are not made in other cells, including that DNA cutting protein. It's called the RAG protein that cuts between these DNA segments. So that's only produced in the cells where this rearrangement occurs. So that is a very tightly regulated mechanism. It happens in B cells, and it also happens in T cells. Yeah.
So is this section of the B cell that gets mutated, is that a particular chromosome or located in a particular place?
Yeah, so there are actually several different antibody genes. There's an antibody heavy gene. So if you look at let's take a look. Yeah, so there are actually two different types of chains in the antibody. There's a heavy chain that's a big segment. I haven't drawn it very clearly here. So there's a heavy chain, and then there's a little light chain that binds to the heavy chain. And you do this recombination process both for the heavy chain and for the light chain. And they're on different chromosomes, and they're actually 2 different possible light chain genes in humans. Yeah. Yeah.
This seems very interesting. Does it create blunt end of sticky?
That's a good question. So the question is, when this protein cuts the DNA, does it create a blunt end or a sticky end? And it actually creates neither. Do we have an eraser? Yeah. So the answer is actually neither of the above. When you start with DNA and it's cut by these cut at these 2 different sites, you actually, the intervening fragment has blunt ends. So there are no bases that are overhanging on either end. But then on the sides, it forms what are called hairpins. So this is when the 2 ends of DNA are joined together. So this end of the DNA strand, this is kind of hard to visualize. This end of the DNA strand is now chemically joined to this end of the DNA strand. And in order for these 2 ends to be joined, there's an enzyme called Artemis, a protein called Artemis, that has to cut at the base of these hairpins. And so now you turn the hairpins into overhangs. And now these can be joined by the joining machinery. Yeah. So it's kind of an elaborate mechanism.
And how many base pairs are cut in this process?
How many base pairs are cut? So what exactly do you mean? Do you mean how big is the recognition site?
When the Artemis cuts off these ends where the DNA strand was connected.
Oh yeah. So I'm not sure the exact number actually. I want to say something like 3 or 4. It's not a lot. It's sort of less than 10. But actually, where this protein Artemis cuts is also somewhat random, and so that can generate additional randomness in the junctions. And in general, these 2 ends, these 2 overhangs, are not going to be compatible, and so they have to be processed extensively in order to be joined together. And so that will generate additional diversity. Yeah. Yeah.
What happens to all the little bits of DNA that get chopped out?
Oh yeah. So actually, the little bit of DNA. So the technical term for these hairpin ends, these are called coding ends. These are coding ends. They're coding ends because they code for a protein. And then these are called signal ends. I'm actually not sure why. And typically the signal ends are joined together. So you end up with these signal ends joined together and it forms a circle, kind of like the DNA that I study in my experiments. And what happens to this little circle? I asked someone who studies this process, and he said that we're actually not sure. It eventually gets lost because the cell divides, and it doesn't get replicated and sent to the other cells.
So is that like a bacterial DNA?
Yeah, so bacterial DNA, yeah, is often circular. Yeah, and that's to solve the problem that when you're replicating DNA, every replication cycle, if you have a linear piece of DNA, you lose some sequence at the ends.
Right.
So human cells solve that problem by putting additional sequences called telomeres at our ends. But bacteria solve that problem by not having DNA ends. So that instead of having a linear chromosome, they have circular chromosomes. But there are bacteria that have linear chromosomes. One way of doing that is to have what's called a prototelomere, which is just a hairpin. So when you replicate this, it turns into a circle, and then there's a protein called a prototelomerase that cuts it in 2 places and forms 2 new hairpins.
So now, presumably, the circular DNA evolved into linear DNA. Is there some advantage to linear DNA?
So one, I had a long discussion about this with a friend of mine. Why do we not have circular DNA? And the answer that we came up with is that it may have to do with recombination in production during production of eggs and sperm, meiosis. So during the production of eggs and sperm, you actually pair homologous chromosomes together, and you can, that's not a very good drawing, let's say 2 pieces of DNA. You pair 2 pieces of DNA together, and you make, this is another place where you want to generate mutations. The cell actually makes double-strand breaks in one of these chromosomes, and that, through a process that I'm not going to draw out because it's kind of elaborate, that can lead to, we're color coding this DNA, that can lead to a recombination event where you get crossing over between the 2 chromosomes. So if you do 23andMe, if you do 23andMe and your mom and dad do 23andMe, you can see which segments of each chromosome came from mom and which came from dad. That's not a very good way of putting it, but yeah. So your chromosomes are all rearranged or recombined in this way every time eggs and sperm are produced. Yeah. Oh, sorry, I actually didn't finish the thought there. And if you had circular chromosomes, then this would kind of make a mess. I'm not gonna try drawing it out, but you basically get, if you had a single crossing over event, you would go from having 2 circular chromosomes to having 1 big circular chromosome, and you'd have to resolve that somehow. You could do that potentially, but that would be really complicated. Yeah.
But then you think that bacteria would want the linear time because they reproduce asexually, so they could get more variation to hit like that.
Yeah, so that's, yeah, so like I said, some bacteria do have linear chromosomes. I think it might be one possibility for why you would opt to have a circular chromosome is that it might be a little bit easier to segregate circular chromosomes at that length scale, because closed circles of DNA naturally tend to kind of come apart. Yeah, it's an interesting question. I don't know if I have the answer to that. Yeah.
Don't bacteria also exchange plasmids too? That maybe that helps with the it's not quite recombination, but it's
Yeah, so that's another interesting point. So Jess pointed out that bacteria also exchange plasmids, which are little circles of DNA, and those can be mobile carriers of information. So even though they don't undergo sexual reproduction in quite the same way that animals do, they can still exchange genetic information using plasmids, and also using something called conjugation, where one bacterium feeds part of its chromosome into another bacterium, and then they can recombine that way. So I kind of misspoke by saying that bacteria don't have sexual reproduction. They do kind of have sexual reproduction, but they do this by means of conjugation, which is a slightly different process. Yeah.
And I think we'll break here, but it doesn't mean you can't ask more questions if you still want to. Just this way, if people need to. Let's thank our speaker again.
Work that I did. So,